Hi, my name is Kyle Stolberg, and today I will be talking about deformed wing virus and how it leads to decreased survival in the honeybee Apis mellifera at increased concentrations. So, deformed wing virus, or DWV, is an RNA virus that utilizes species of Varroa mites as its main vectors. In the absence of mites, DWV is transmitted to other bees via an infected queen's ovaries and via royal jelly secretions from infected nurse bees. As a result, the virus is present in most apiaries around the world and can have profound adverse effects on the ability of adult bees to survive infection. DWV is most commonly known to infect the honeybee species Apis mellifera, which is the most common honeybee species worldwide. This makes the species Apis mellifera the ideal model organism, both because of its integral role in honey production worldwide, as well as being the species most commonly affected by the virus. DWV affects primarily the heads, abdomen, and wings of infected bees. This can lead to morphological deformities such as shrunken and deformed wings, which is what the virus is named after, but also leads to additional symptoms such as shortened and rounded abdomens, paralysis of the legs and wings, and impaired cognitive functions. Furthermore, particularly in the presence of infected Varroa mites, DWV can result in the immunosuppression of infected bees, which increases the bee's susceptibility to other opportunistic pathogens. This leads to the question, how does deformed wing disease affect the survival of adult honeybees? Because infected bees display a wide variety of phenotypes related to DWV, scientists wanted to look at differences between viral titers amongst adult bees and the severity of phenotype. The groups observed were uninfested non-deformed bees, infested non-deformed bees, mites from non-deformed bees, infested dead and deformed bees, and mites from dead and deformed bees. Focusing in on the bee groups, the uninfested non-deformed bees had the lowest optical density or viral titer, followed by the infested non-deformed bees, and then the infested dead and deformed bees, which had the highest optical density and therefore the highest viral titer. This suggests that as severity of phenotype increases, so from phenotypically normal to death from infection, viral titer increases, suggesting that increased concentration of DWV leads to higher mortality and decreased survivability of adult bees. Furthermore, this suggests that a certain threshold concentration of DWV must be reached before the virus becomes fully pathogenic. Since Apis mellifera are found all over the world and are most commonly affected with this virus, scientists also looked at the role DWV played in observed colony collapse. So 29 colonies showing signs of infection of DWV were observed over winter to observe whether or not this infection resulted in colony collapse. During this time, the approximate proportion of worker bees exhibiting DWV symptoms were recorded for each of the colonies. So of the 29 colonies, it was observed that 16 of the colonies survived, whereas 13 of the colonies collapsed. In the colonies that collapsed, there was a significantly higher proportion of adult bees that exhibited signs of DWV, so around 72%, compared to the colonies that survived. This suggests that as more adult bees are infected with DWV, the survivability of the colony decreases. Because Varroa mites are the main vector of DWV, scientists wanted to determine whether or not Varroa resistance would increase the survivability of adult bees infected with varying concentrations of DWV. To do this, they compared 3 strains of Apis mellifera, labeled IHP, which was Varroa susceptible, and then POL and RHB, which were both Varroa resistant. Furthermore, the strains of bees were fed varying concentrations of a DWV lysate, which was obtained from worker bees infected with DWV. The 5 treatments were without feeding, which was the negative control, fed PBS only, an additional negative control, fed DWV lysate without dilution, denoted D1, fed a 100-fold dilution of lysate using PBS, which was denoted D1 in 100, and fed a 1,000-fold dilution of lysate using PBS, denoted D1 in 1,000. The scientists then measured the average days of survival after treatment for each of the 5 treatments in each of the 3 strains. In the Varroa susceptible strain IHB, the negative control group without feeding showed the longest average survivability of about 12 days. This was followed by the negative control fed only PBS, which had an average survivability of 11.7 days. Conversely, adult bees given DWV lysate showed decreased average survivability compared to the negative controls, with the D1 group surviving about 9.8 days, the D1 in 100 group and D1 in 1000 group surviving about 10.75 days. In the Varroa-resistant POL group, those given the negative controls once again had a higher average survivability than those given the DWV lysate at D1 and D1 in 100 concentrations. However, the D1 in 1000 group showed a survivability intermediate between the 2 negative control groups. Conversely, the RHB group that was unfed lived the shortest, and the groups fed PBS and 1 in 1000 DWV lysate lived the longest, followed by D1 and D1 in 100. This data suggests that DWV decreases the survivability of adult bees that are Varroa susceptible and likely decreases the survivability of adult bees that are Varroa resistant at higher concentrations. In conclusion, as the virus titer of adult bees affected with DWV increases, so does the severity of the phenotype. Therefore, as the virus titer increases, the survival of adult bees decreases. Furthermore, of the colonies affected by DWV, that approximately 45% of the colonies collapsed, further emphasizing the decreased survivability of adult bees following infection. Additionally, even though Varroa mites are the main vectors for DWV decreases the survivability of adult bees in both Varroa susceptible and Varroa resistant honeybees. This research emphasizes the significant adverse effects of deformed wing virus on adult honeybees, particularly in Apis mellifera. This research shows the dangers of DWV and the role it plays in decreasing the survivability of adult honeybees. Furthermore, it emphasized the importance of beekeepers protecting colonies from DWV infection, or else it could result in colony collapse. Finally, the research encourages additional research on viruses similar to DWV and possible therapeutic treatments that could be used to treat this virus.